for it to be able to show up in a dispenser sample. So up to this point, we're about an hour and a half into this. There is light at the end of the tunnel. But at this point, we know a lot more now about microbes and how they grow and what they do and how they do it. And we know a lot more about the importance of good sampling techniques than we did before. Now that we have our samples, okay, we pulled the samples, we followed good sampling protocols, we now have to transition away from, is this going to tell me what I want to know? Or is this the right kind of sample to tell me what we want to know? We're transitioning into getting that sample to tell us the right story, so to speak, which means we have to ask the right questions. You can't have the right answers if you don't ask the right questions. And from a fuel microbiology standpoint, from a scientific standpoint, those questions have to be along the lines of, do I have microbial contamination above a certain level in my storage tank? How much microbial presence do I have in the tank? And then third one, is the presence that I do have, is that contributing to some existing problems that I may know about? Or, if not, how likely is it to do so in the near future? Now, these three questions seem to be interrelated in some way, but they're not all answered in exactly the same way. This means that we really need to know the right testing approach to take if we're going to best answer these right kind of questions. And these are the kind of considerations that go into what fuel microbiologists refer to as condition testing. See, we have transitioned from condition monitoring into what is now condition testing. So there are 4 broad categories for condition testing. I know it says condition monitoring on there, but that really should say 4 categories for condition testing that we should consider. There's gross observations, there's physical tests, there's chemical tests, which oftentimes get paired with physical. So if you were to say there's 3, you'd say gross observation, physical and chemical tests, and then the last one, which is not by any means least, microbiological tests. So what we're going to do now is we're going to delve into these categories. We're going to talk about what kinds of tests go into those categories and what the roles or what values these tests may have towards helping us to answer those right questions that we seem to be asking. All right, so let's start with the first one, gross observations. Start at the simple end, the observations that we can make with our 5 senses. Now, really it's the 4 senses because we don't really recommend taste here, but keep it simple, the 5 senses. So we get a fuel sample. Now we're going to assume that it's a good fuel sample. We're going to assume that we have done our best to get ourselves a good, viable fuel sample. So we look at it. We look to see if it looks different. You could smell it. Are there characteristic odors? You pull a fuel filter media. You pull a fuel filter or a tank filter, and you look at the filter media, and you see a coating on there. Most of them are going to have coatings, but what does that coating feel like? Does it feel like kind of a dry particulate, or is it kind of slick and slimy? Those kind of things. Those all go into gross observations. Now, when you're visually inspecting fuel, we said look at the fuel to see if it looks different. When you're visually inspecting fuel, well, there is an ASTM protocol for that. It's D4176, visual inspection for water and particulates. And that's actually a 2-step process that can really help someone, I don't want to say streamline this, but do this in a rigorous manner. So the first step would be you look at it and you check for free water and particulates. Now that's provided that you don't already have a clearly visible water phase. Because if you do, that answers the question right there. So what you would do, I mean, there is a protocol you would do for this. What you would do, you don't just take the sample and you hold it up in the jar and you say, it looks clear. No, what you do is you've got it in a sealed container and you swirl it vigorously. You swirl it hard for about 30 seconds, and then you hold it up to the light. And now what you'll see a lot of air bubbles that are swirling around in it. Excuse me. And those air bubbles will start to rise to the sample surface. But any water and any particulate that's in there will actually start to sink to the bottom. Then what you do is you compare the clarity of the fuel sample and you compare it against the visual chart that comes with D4176. And there's some examples of it there on your screen. It's got those lines, and that actually enables you to give the fuel a numerical clarity rating between 1 and 6, with 1 being clear and 6 being not. So if the stored fuel doesn't pass this visual clarity inspection, well, that right there, is a pretty excellent indication that there is something amiss from the fuel. This is the simplest test for it to run, and if it doesn't pass this, then it's pretty clear that you've got problems. Now, physical and chemical tests. We have said before that microbial contamination changes the properties of the fuels in certain distinct ways. Ways like it can increase the total acid number, it can increase the peroxide number, it can make it hazier. Remember the Zwitter ions and the way that they create invert emulsions. Well, that makes the fuel hazy, increases the haze. Increases the amount of total suspended solids. It can reduce cetane rating in diesel fuel. That's a big one. We already mentioned, in gasoline, because of the consumption of the lower end carbon molecules, you can change the vapor pressure. So it can have those and other detrimental physical changes to the fuel. And so there are certain tests that we would use to document and measure the physical and chemical properties of the fuel, as well as try to measure the properties of the fuel that might predict whether it will change in the future. Now, in this case, having said these, the most important tests that we would look at would be as you see on your screen, the stability tests. Accelerated oxidative stability, accelerated thermal stability, and then the one below that, petrooxy. Petrooxy is actually the preferred stability test for ultra-low sulfur diesel fuels. Now, if you've gone through the stability training that we've done in the past, and if you're just watching this on the Bell YouTube channel, you can find that in the playlist. When we talked about stability in the stability training, what we tried to get across is that stability tests are predictive tests. Stability tests try to predict whether the fuel sample will change in the future. Because if the fuel's in storage and something has happened to it, like let's say there's been microbial growth and you've had acids that are produced that have started to degrade the fuel, if something has happened to the fuel while in storage, then the fuel is more likely to change condition at some point in the given near future. And thus, physical stability tests like those three can give us, or can help us to have more of an analytical expression of that likelihood for the fuel to change. Now, we can also do other physical tests to assess the physical state of the fuel. And those don't just simply assess it in a vacuum, but it also can assess it with respect to judging whether these tests can infer microbial contamination too. I mean, that would be helpful, right? What tests can help infer the presence or not of microbial contamination? Well, we've got 3 ones on there. There's a filter blocking tendency test, D2068. This helps determine the particulate load of a fuel, and if you've got a microbial contamination, the fuel by nature is going to have a higher particulate load. Hence, 2768 infers possibly microbial contamination. Kinematic viscosity, 445. That's a pretty basic test. And in principle, it is important to verify the viscosity of any kind of fuel that you're using because viscosity is a big factor in whether how it behaves when it's being injected, especially in high pressure common rail engines. And since we know that microbes can consume smaller hydrocarbons in a fuel mix, over time that will change the viscosity of the fuel. And if it changes it too much, then you're going to have problems. So you can use kinematic viscosity test to help infer microbial contamination. Water separation properties, 7451. That's not really a test we have talked about that much, but 7451 tests the effects of microbial contamination on water separability of the fuel. Remember, microbes produce those Zwitter ions, those biosurfactants. And the biosurfactants make the fuel hang on to the water. They produce the invert emulsions. So if the fuel has microbial contamination and it has produced these biosurfactants, you will get an emulsion formed in the test that is not going to be easily resolved. Other tests we might consider, which we would split up or separate, categorize between tests done on the fuel phase and tests done on the water phase. First, test done on the fuel. You've got Carl Fisher water content. You've got peroxide number. You've got fuel corrosivity, total acid number. And then test done on the water, pH. Always best to do pH on the water phase, and total dissolved solids in the water. So we've covered gross observations that can hint at microbial contamination, and we've covered the physical and the chemical tests that can give us a more measured confirmation of the properties of the fuel, which might hint at something has changed the fuel's properties. And given the tests that we choose to look at, we know that those fuel properties change most likely. If they have changed, it's most likely because there's been microbial contamination. So the last category of tests that we would undertake are the ones that are most applicable, most specific to measuring or detecting microbial contamination itself. And those are, of course, the microbiological tests. So let's talk about those because there's plenty to talk about. There are three main types of microbe detection tests that we would consider. This does not count the ultra complicated genomic tests where they break, it's done in a genetics lab, and those are actually the most accurate. But when it comes down to it, they're rather specialized and they're rather expensive. So for the real world, for those of us who do not have access to a genetics lab, we have three different main options. And the different options for microbiological testing, as we discuss those, this should illustrate to us an important point about how the right or the wrong test only gives us really a fraction of the picture. Really, they all only give us fractions of the true situation. The question is going to be how big of a fraction are we going to get? So the first ones we look at, culture tests. D6974 and 7978. Culture tests are the oldest microbe tests. They date all the way back to the advent of germ theory in the middle of the 19th century. And culture tests have limitations that by nature, they only detect a very small fraction of what we would call the microbiome. Microbiome being the entire microbiological picture of a given environment, in this case, a fuel and water sample, which actually comes out of a bigger microbiome, which is the storage tank. Culture tests only detect like 1/10 of 1%, 0.1% at best. Why do they only get such a small fraction? 0.1%, that's nothing. Why do they get such a small fraction? Well, when it comes down to it, a culture test depends on the ability of cells to grow under the culture conditions, which likely are very different from the environment from which we took them out of. And so when you're doing a culture test, you have two kinds of variants that can happen here. Now, there are, in addition to active cells, that are the ones that are active and growing and the ones that are ideally going to be measured, there's also two other kinds of cells. There's also injured cells, and there's also dormant cells. Injured cells, of course, pretty self-explanatory. Microbial cells that, for some reason or the other, are injured and they're not really going to grow. They're not really going to reproduce on a culture test. And if they don't reproduce, you're not going to detect them, right? Second one, the dormant one. Dormant ones, again, they're not active. They're not going to reproduce. And so now, let's take a step back here, though, because you say, well, if they don't reproduce in a culture test, that means they're not reproducing in the storage tank. But the distinction here is that you can you're taking a dormant cell, you're taking it out of the tank, you put it in a culture medium, and hopefully they start to grow. But you could have any number of those cells that were active. They were active in the storage tank, but you put them in the culture medium, they won't grow. Those are the ones that become, essentially, they fall out of your final results. They could become dormant. The bottom line is that just because cells are dormant in the culture media does not mean, by any means, that they were dormant in the tank. And that is why, one of the reasons why you can have such a significant difference. So those dormant, those injured cells, they don't show up on the culture test, but they were alive and kicking in the storage tank. So the first problem was you only get a really small percentage of the microbes that you're detecting. Second one, which is a huge issue for accuracy, is potential for contamination. Culture tests rely on us being able to grow a representative sample of the microbiome and to grow it in the specific culture medium. That's what it takes for it to be a viable test. Now, individual microbes way too small to be seen by the naked eye. In order for a colony to be visible, it has to have about 1 billion cells in size. So if you see a little moldy spot on your bread, you probably got 1 to 10 billion of fungus cells on there. So it takes 1 billion cells for a colony to be visible. Now, remember, you're growing these from scratch, essentially. It takes 30 generations of reproduction for one cell to become 1 billion cells. So we're waiting on this. We're waiting on these 30 generations to happen while the test is incubating, because it takes an incubation period. That's another one of the disadvantages. You don't get instantaneous results because you have to wait for those results to develop. So let's say that you start in your original thing. Let's say that you start with 8 original microbe cells. Okay. You got them in your culture medium. But let's say that in addition to those 8, you have 2 microbe cells that came in from the outside. You had cross-contamination. You now have 2 microbe cells that were not present in your fuel sample, and thus they were not present in your storage tank. So you've got 2 out of 10. Now, you let them incubate. They're all multiplying. But remember, though, that not all microbes grow at the same rate. Some grow fast, some grow slow. So what happens if, say, those 2 hitchhikers are the kind, let's say they grow twice as fast as the other cells. So you let that happen over 30 generations, what are you going to have? Well, you're going to have a whole bunch of microbes. You're going to have 1 billion microbes. But how many of those are the ones that were actually in your sample to begin with, and how many of those came from those 2 cells at the beginning that grew twice as fast? I don't know what the mathematics of it, but what I can say is that there is significant potential for your culture results to be significantly different than what the picture of your actual sample and your actual tank should really look like. And how do you know which of those billion cells came from those cross-contaminant ones and which ones actually came from your storage tank? You really don't. So, and that can be a problem. All right, so there are that's basically how culture tests work and what some of the big problems with them are. But there's different kinds of culture tests. There is, so let's talk about a couple of them. First of all, you see membrane filtration. That's actually colony count membrane filtration. That is D6974, and that is the laboratory standard test. What they do is they take a test sample, they filter it, and by test sample we're talking about like fuel that might have microbes in it, planktonic microbes. They filter it through a membrane that is actually filled or inoculated by culture medium. So it catches the microbes. The microbes stay there, and you incubate the membrane. You allow it to grow for a certain period of time. And so hopefully you get a picture of what's going on in your tank. So that's membrane filtration. That's the laboratory standard. There's also Microbe Monitor 2, D7978. Now, that is what's being pictured by that guy in the blue latex gloves. It's a glass bottle, and it has this stuff they call thixotrophic gel. Thixotrophic gel refers to gel that actually moves faster when you shake it than when you shake it slowly. And it's a culture medium, and so you inoculate it with a sample of fuel or whatever, and you leave it, and it actually grows microbes, and you can see colonies that show up, and you can count the number of colonies. Then you can compare to a chart, an interpretation chart, and it allows you to do somewhat of a quantitative estimate for the number of microbes. There's also liquid culture mediums, which are those glass jars that you see with all varying shades of color. Microbiologists would also refer to liquid cult as non-selective broth. And the broth actually contains a dye that actually, as microbes grow and they produce acids, it detects that acid and it can turn red. How red it is correlated to the number of microbes that are in there. And so you can get kind of an interpretation of the number of microbes in your given sample. And then there's the last one, BART. BART ones are kind of similar to liquid cult. They're packages of multiple bottles, and they actually enable you to grow specific kinds of microbes. And what that enables you to do is it enables you to be a little bit more specific on exactly what kind of microbes are growing in there, which could be useful if you're trying to figure out what to do. I mean, one of the bottles selects for sulfate-reducing bacteria, which can cause serious problems. Some of them select for denitrifying bacteria. Some of them select for acid-forming and slime-forming, and there's a bunch of different kinds of bottles. So in the end, depending on the results that you get, you can get a more specific picture of the kinds or the classes of microbes that are in your sample, which it does give you a little bit more useful information. Now, last important question to consider with culture tests, and we have alluded to this already, is how long do these take? This is important, and we've mentioned it, but we haven't really considered it in depth. And it matters because it can affect our test results. Again, remember, not all microbes grow at the same rate. Some microbes will split every 30 minutes. Some microbes will split every 12 hours, which is a lot slower. The ones that every 30 minutes, they can produce a visible colony in like 16 hours. The ones that split every 12 hours, well, they're going to take like 2.5 weeks or more to produce a colony. So if you've got a culture test and you wait 3 days for it to incubate, how do you know if what you're seeing really does represent your microbial biome? If a significant number of those microbes that came in the sample are still in the process of growing, you don't really know, and you don't really have a meaningful way of knowing either. And so that kind of limits the usefulness. Excuse me. All right. Then you've got these things called LFDs, lateral flow devices. Now, lateral flow devices are fancy names for what scientists would call immunoassay tests. Now, immunoassay seems like a big word. Assay, split it up into immuno and assay. Assay refers to measurement. Immuno or immune refers to microbes. So an immunoassay test would be a kind of a test that's able to measure the amount of small molecules, like bacteria, measure the amount of small molecules in a solution using some kind of antibody substance that detects it. So the most popular lateral flow device is the Fuel Resonate test by Canidia. I think Canidia Biosciences. And it is got an ASTM number, 8070. And each test is actually a panel of 3 tests. They're all on a cardboard backing, and they've got 3 pairs of these, what they call LFD modules, that you take fuel, you prepare fuel with their reagent, and then you put drops of it in there. And what it's supposed to do is it's supposed to detect, or it's supposed to give you an indication if you've got low or medium or high contamination levels of 2 different classes of microbes, bacteria and fungi. Now, at its best, Fuel Resonate can detect about 60% of the microbes, which is a huge improvement over culture tests that are like 0.1. It's 600 times more accurate, so that's good. But there are downsides to it that should give one pause if one's going to use Fuel Resonate as your primary indicator. It kind of tells you low, medium, high, but it does not give you a specific number. It also does not give, it gives quick results, not as quick as some others, but quicker than the culture test. But it's not able to be calibrated. That's a big one. Just as you ask of a culture test, how do I know if these are accurate? How do you know if you have to store these LFD tests under controlled conditions? Well, how do you know, like, if you left it in your car for a certain amount of time or in the technician's truck and he does the test, how does he know that the test is still viable? There is no way to know for sure. And that's a problem because the last downside is it's expensive. It's like $150 a shot. So if you're if you're using a bad test and you don't know, you have thrown $150 down the drain, which I don't know about you, but $150 can buy me a lot of hamburgers or something. So yeah, that's not really good either. This brings us to the last test, the one we're going to talk the most about because it's the last one and it's the best one. It's one that we're the most concerned about, ATP testing, D7687. Now, you have to understand a little bit about the history of ATP testing because ATP testing was first developed in the 1950s. Okay. Now, the testing we do now is a whole lot better than the tests we did then, but it's not something that was developed in 2010 or something. ATP testing is based on the principle that all microbial cells, really all living cells, we should expand it. All living cells have a given amount of ATP in them. ATP is present in every living cell because ATP, adenosine triphosphate, is the molecule that's involved in energy production and transportation. So every living cell has to have ATP. So what the 7687 test method does is it calls for us to isolate the ATP from the microbial cells that are in the fuel sample so that we can measure them. Now, the basic process, I've done this test 100 times. I'm better at it than I was at the beginning. It's really not the most complicated thing that you would ever have to do, but it is something that takes a few times to master. which is to say that any technician could master it if they put their mind to it. Here's the basic process here. Okay, you have a fuel or water sample. You take a given amount of it and you run it through a filter, little filter. The filter takes out all the microbes, plus a bunch of other stuff that's in there. Microbes plus impurities, we'll call it. Okay, so you got microbes in there. You've also got impurities in there, and you need to get rid of the impurities because you just want the microbes. So you run a solvent, a cleaner solvent, through the filter, and essentially it washes out everything but the microbes. Then you dry it. You run a couple bursts of air through it. It dries it, basically. And then the ATP that's in the microbe cells use another liquid, and that liquid actually causes the microbial cells to actually rupture and burst open, and all of their ATP gets extracted out of the filter, and it's extracted into this tube of diluting liquid. That's basic step 3. So now you've got a bunch of ATP-filled dilution liquid, and so you take a little bit of that, and you combine, put it in a test tube, and then you take a little bit of this special enzyme called, one trade name from Luminultra is called luminase, but you add this enzyme to it, and you mix it up. And what the enzyme does is that when you expose that mixture to UV light, it causes the ATP to glow, to produce light, essentially. And so you take that test tube, you put it in this luminometer, which is a specialized piece of equipment, and the luminometer basically can measure, expose it to UV light, and then measure how bright the glow is, and it will be able to calculate for you exactly how many microbes were actually in that sample based on the amount of ATP that it was able to detect. So the culture test detects 0.1, 1/10 of 1%. The LFD, remember the lateral flow device, detects about 60% on its best day. ATP testing will detect about 70% of the microbiome, which means it's 700 times more accurate than a single culture test. Now, not only is it much more accurate, but there's other significant advantages that really make ATP testing really, for lack of a better way to put it, the preferred way to do things. It's quick to perform. Like I said, I've done 100-something of these tests. Any technician trained on this and gets to do it a few times could do it in the field, and they could complete a test in less than 10 minutes. Less than 10 minutes, you know exactly how many microbes are in there, and what that means is that you're not waiting for 3 days. You're not waiting like you had to do 10 years ago, which is 28 days for results to come back from a lab. You're getting the results in 10 minutes. What should we do? We have this many microbes. Boom, that means we should arrange for cleaning or apply biocide, or we don't need to do anything. I mean, it enables real-world, accurate, good decisions to be made. And that comes from the fact that it is fast and it is accurate from a quantitative standpoint. Now, it does not detect dormant cells. That's the one downside. But that's really the only trade-off with it, and it's not really a significant enough trade-off for us to say that ATP is not the best thing that we could be doing. So that's the ATP testing for condition testing, confirming the condition of the test. Now, one of the takeaways from talking about all this, we have touched on this before. One of the takeaways from all this is that it is important for us to keep a proper perspective on the accuracy of the tests that we get and what the real meaning of the microbe tests are that we've gotten. This is because a positive or negative test may not tell us the entirety of what we think it is telling us. Now, for example, if you do a culture test dip slide and it comes back negative, do you know for sure that that means you really have no microbes in that tank? Well, if the answer is no, then what are the possibilities for why it doesn't tell us that? Well, since the culture test is done on a fuel sample, if the sample was not, really representative of the conditions in the tank, then the sample was wrong, and hence our test was wrong. Or it could be possible that, because the culture test might only detect a small number of microbes, maybe the culture test wasn't specific enough to show us the actual amount of microbial presence. In the end, negative microbial results can mean one of three things here. It can mean either that we have the wrong sample. That means we really did have microbes in the tank, but our sample didn't capture them. It was the wrong sample. It could mean wrong test method. Microbes were actually in the sample. We did get a good sample, but the test method did not detect them. Or it could mean that we actually have the right sample and the right method, which means if we have a negative test result, if it's right sample, right method, the system actually is uncontaminated and our negative test result is valid. Now, so you have to keep that in mind. Now, we also, with respect to ATP testing, there are some nuances that we have to remember even when we're getting those kind of test results. With ATP testing, typically, depending on whether you test fuel or water, you'll get a microbial equivalent number. And then, again, depending on whether it came from fuel, then if it's above, X, if it's above 10, then you're going to get a red result. If it's between 1 and 10, you're going to get a yellow, which is, it's kind of like the yellow caution light. It means you need to start paying attention. If it's below 1, it's green. Now, if it's water, you've got a different scale because you need more microbes in water to indicate a problem. But you're going to get raw numbers, and then they're going to be interpreted. Red, yellow, green. Not a problem, might be a problem, or severe contamination, you need to do something. But so red, yellow, and green correspond to ranges of total microbes that were detected by the ATP test. And the colors used are intended to imply if contamination is moderate or if it's serious enough to warrant a specific kind of action. However, in the interest of knowing, trying to understand what the test is really maybe telling us, this is only total microbe count that we're getting here. The ATP test is giving us a total microbe count. But not all microbes are equally problematic, right? I mean, remember earlier we talked about having, manageable levels of microbes as kind of our working goal. We said that whether a given level of microbes was a problem, we said it would depend on what the levels were. And we already know that 1 million microbes of one kind, one species, one type, may not be equivalent from will it cause a problem standpoint to having 1 million of another kind, or even 10,000 of another kind, or 10 million of another kind. It all depends. And so not all reds, and not all greens, and not all yellows are created equal. Depends what's in those greens and those reds and those yellows, the kinds of microbes. Unfortunately, though, we don't usually have the ability to get as granular as that. So we have to go with what we have the capability of looking at, which is total microbes. We just keep those other facts in the back of our minds just in case we do not get the expected results that we were expecting. So that is a brief consideration of the main microbe testing options that we have. Our goal has been to detect and measure if we have microbial content present. That's what our goal would have been when we undertake one of these tests. Based on the results from those tests, whatever tests we choose, We then have to transition from condition monitoring and condition testing. We now transition to the last part of this. 2 hours and 10 minutes later, we are transitioning to contamination control, which involves controlling and getting rid of the problem that we think we have. Now, we consider that everything that we have talked